Do you remember the movie The Abyss of James Cameron from 1989? In the movie, these creatures in the deep of the ocean are aliens, and I always saw that way. But after I studied biology and know ctenophores, in my mind, these creatures are just ctenophores that evolved in the deep of the ocean like the humans evolved inland. While the human evolved through 50, 60 million years, the ctenophores could evolve through at least 500 million years. So the next time you watch this movie, think about it. The first recognized figure of a ctenophore is a draw made by a doctor on a ship in 1671. Linnaeus and Cuvier put them among the zoophytes. In the 19th century, Schultz proposed the first rational classification to medusas and pelagic ctenophores, creating the order Ctenophore. For the ctenophore, discophore for all solitary medusas and cnidarians, and siphonophore for all cynophores and colonial chondrophores. He considered these orders part of the class Acalepha as a group between zoophyte and echinodermata. In 1889, Hotchek take off the Ctenophorans from the Acalepha, creating their own class, today a phylum. Like Cnidarians, Ctenophorans has radial symmetry, with exceptions. They are distinguished from all other animals by having coloblasts, which are stick and adhere to prey, although a few ctenophore species lack them. The outer surface bears usually 8 combi rows, called the swimming plates, which are used for swimming. The rows are oriented to run from near the mouth, the oral pole, to the opposite end, the aboral pole, and are spaced more or less evenly around the body, although spacing patterns vary by species, and in most species, the combi rows extend only part of distance from the aboral pole towards the mouth. The combs, also called the kidneys or comb plates, run across each row, and each consists of thousands of unusually long cilia, up to 2 millimeters. Unlike conventional cilia and flagella, which has a filament structure arranged in a 9 more 2 pattern, these cilia are arranged in a 9 more 3 pattern, where the extra compact filament is suspected to have a supporting function. The largest single sensory feature is the aboral organ, at the opposite end from the mouth. Its main component is a statocyst, a balance sensor consisting of a statolite, a tiny grain of calcium carbonate supported on 4 bundles of cilia, called balances, that sense its orientation. The statocyst is protected by a transparent dome made of long, immobile cilia. A ctenophore does not automatically try to keep the statolite resting equally on all the balancers. Instead, its response is determined by the animal's mood. In other words, the overall state of the nervous system. For example, if a ctenophore with trailing tentacles captures prey, it will often put some combi rows into reverse, spinning the mouth towards the prey. About the digestive system, when prey is swallowed, it is liquefied in the pharynx by enzymes and by muscular contractions of the pharynx. The resulting slurry is wafted through the canal system by the beating of the cilia and digested by the nutritive cells. Two pharyngeal or paragastric canals recur and run parallel to the pharynx. Two transverse channels start at the right angles to the stomatal plane and divide into three other branches. The median branch of each crack, the tentacular canal, leads to the base of the tentacular sheath. Each of the other two branches, the interradial channels, forks to form a total of 4 adradial channels on each side of the animal. These, in turn, connect to 8 southern channels, one under each row of combs. Finally, a number of channels passes from the infundibulum to the alveolar pole, where it divides below the apical sense organ into 4 short channels, 2 ending in a blind bottom and 2, the anal channels, opening to the outside by small pores called anal pores. The anal pores serve as a primitive anus, assisting the mouth in defecating indigestible waste, also serving as an outlet for metabolic waste. In Ctenophore, bioluminescence is caused by the activation of calcium-activated proteins, named photoproteins in cell called photocytes, which are often confined to the meridional canals that underlie the 8 comb rows. The ctenophores are divided in 2 classes. Class Nuda has one only order, Beroida, and just one family, Beroidae. They are distinguished from other comb jellies by the complete absence of tentacles in both juvenile and adult stages. Species of the family Beroidae are found in all the world's oceans and seas and are free swimmers that form part of the plankton. Examples: Neis cordigera, Beroe abisicola, Bere cucumis, Bere ovata. Class Tentaculata is a class of comb jellies. The common feature of this class is a pair of long, filtry, contractile tentacles which can be retracted into specialized ciliated sheaths. In some species, the primary tentacles are reduced and they have smaller secondary tentacles. The tentacles have coloblasts, which are stick-typed cells that trap small prey. The class Tentaculata has 8 orders. Order CYDIPPIDA is an order of comb jellies. They are distinguished from other comb jellies by their spherical or oval bodies and the fact that their tentacles are branched and can be retracted into pouches or either side of the pharynx. The order is not monophyletic, that is, more than one common ancestor is believed to exist. Examples: Aulacocitena cydippidi, Platyctena species, Dryodora glandiformis, Euplocamis dunlapé, Requelia rubra, Ormifora plumosa, Pleurobrachia pileus, Duobrachium sparxé. Order LOBATA is an order with smaller tentacles than other ctenophores and distinctive flattened lobes extending outwards from their bodies. Examples: Batocire fosteri, Bolinopsis infundibulum, Bolinopsis vitrea, Mnemiopsis leidyi, Deiopia caloctenota, Eurampai vexiligera, Lampochites cruentiventer, Leucotia pulcra, Lobatolampia tetragona, Ossiropsis crystalline, Ossiropsis maculate. Order Platyctenida. Is the only bentonic group of organisms in the phylum Ctenophora. Platyctenida are considered to be a phylogenetically young group along with the orders LOBATA and Beroida, and are believed to have stemmed from an ancestral version of the order CYDIPPIDA after some kind of a bottleneck effect in the phylum. Examples: Celoplana astericola, Celoplana meteoris, Valicula multiformis, Ctenoplana Kovalevsky, Liroctes flavopallidus, Liroctes imperatoris. Order Ganeshida. They are characterized by pair of small lobes round the mouth and extended pharynx. Two species are currently recognized: Ganesha elegans and Ganesha anamita. Order Cryptolobiferida. Family Cryptolobatidae. Example: Cryptolobata primitive. Order Thalassocalida. Combi jellies known from the California coast, Gulf of Mexico, and West North Atlantic. It is represented by a single species, Thalassocalice inconstans, which is the only species in the family Thalassocalcidae. Is a pelagic ctenophore typically occurring in upper mesopelagic depths, but has been observed at depths up to 3,500 meters in Monterey Canyon. Order CESTIDA. Unlike other combigellies, the body of Cystids is greatly flattened and dry out into a long ribbon-like shape. The two tentacles are greatly shortened, and two of the four ciliate combi are reduced. The unusual body form allows the animals to swim by means of muscular undulation, as well as by using their cilia. The largest species, belonging to genus Cestum, can reach 1 meter in length. Examples: Cestum veneris, Velamen parallelum. Order Cambogida. I failed to find some important information about this order, except the mention in the sources.